Great, so let's get started on time. Hi, welcome everyone. Thank you so much for taking the time to attend this this webinar, which is a series of webinars that EBTC has started producing 2 years ago. Paul, could you go to the next slide, please? Great. So the topic of the webinar this time is ontologies for toxicology and environmental health. Next slide, please. So the topic is one of the main topics of EBTC. So what is EBTC? If you haven't heard about us, we are an international organization, a collaborative effort started over 10 years ago at Johns Hopkins School of Public Health, focused on improving the standards in toxicology and helping make toxicology into a transparent, objective science. So our efforts are focused on 4 major topics, which is raising research standards, making sense of evidence, improving access to research, and advocating evidence-based decision-making. All of these topics are, some of them are very tightly related to systematic reviews and systematic methods. But today we are talking about improving access to research and how ontologies and data is helping us get the evidence, or what are the deficiencies in how we put together and assemble and present and publish the data, and then bring it into evidence. What are the gaps, and how can we fill them? So next. So some of the examples of what we do at EBTC and some of the projects that we've done over the last few years is such as applying systematic methods, including systematic scoping reviews and systematic reviews to adverse outcome pathways, working with editors to improve publishing standards around systematic reviews, evaluating non-animal test methods, mechanistic methods for drug-induced liver injury, and out today, actually updating Bradford Hill's concept of biological plausibility for modern systematic reviews in environmental health. So take a look at that. Next. So for full disclosure, these are the funding sources of EBTC. We are funding for Center for Alternatives to Animal Testing at Johns Hopkins School of Public Health. And we have a lot of collaborators who are contributing to our project. So this is in-count contributions that we would like to acknowledge. The big one is US EPA, US FDA, the Tox Strategies, Norwegian Institute of Public Health, and SIOM. So thank you. And finally, we would like to encourage everyone to join the collaboration. We are operating on open Cochrane-like model, so anybody could join and become a member of EBTC. We are in the process of launching our network, the new network structure. For now, you can just go onto our website and subscribe to the newsletter, and more news will be coming soon. We will put the links in the chat on how to join EBTC during the webinar. And if you have any questions, you can, of course, contact me or Paul. And so here I'll give the rights to continue with this and lead this discussion to Paul Whaley, who is a research fellow at EBTC. On to you, Paul.
Good stuff. Thank you, Cassia. Good. Thank you, everyone, for coming. It's great to see so much interest in what I think to many people is quite an abstract topic, right? So we're talking about ontologies in toxicology and environmental health today. I have 3 great speakers for you. We'll be talking about, really it's kind of a primer, I guess, is the idea. So we're talking about computable publishing, the role of ontologies and taxonomies, metadata in computable publishing. And we're going to be talking about what they are, how they're made, and then how we can make use of them. Of course, probably asking why we're talking about ontologies at all. The reason kind of goes back, not all the way to this particular molecule here, but this molecule here has had a big part in helping people understand why we need better data sharing methods in research. So bisphenol A was a real poster child of some of the challenges we're having in chemical risk assessment, chemical risk management, kind of in the mid-2000s, right? So it's a very useful molecule because it helps make polycarbonate plastics hard, and it's really good for making resins for lining food cans. And also was widely used in thermal ink in receipt papers and things. The problem was, because of its utility, human exposure to this ended up being more or less ubiquitous. So if you looked for bisphenol A in people's blood, you would always find it. And this started to become a concern because bisphenol A started to look like it might be acting in the body as a xenoestrogen, which made some regulators a little bit worried and NGOs a little bit worried. And lots of research started to be done. By sort of 2019, 2020, we had lots of evidence, for example, of how bisphenol A might be impacting human fertility. But we also had lots of studies on lots of other endpoints as well. So we had people concerned that it might be impacting gender dimorphism, might be a risk factor in obesity, might be a risk factor in premature birth and breast cancer and behavioral disorders and premature puberty, and the list kind of goes on and on and on. So we have a ubiquitously exposed population with multiple health outcomes of concern, right? Now, of course, not all these risks are real. Some will be more pronounced than others. We want to regulate based on good understanding of the hazards and risks posed by exposure to a chemical substance. And the best methods we have these days now for determining which risks are real are, of course, systematic review, where we locate, evaluate, and synthesize all the available evidence relating to the health risks posed by a chemical substance. The problem we have with systematic review is it typically involves searching and screening between 4,000 and 40,000 references. It will be detailed analysis of between, say, 15 and 150 studies. It takes about 18 months to do a smaller systematic review, where we're talking about screening 4,000 to 10,000 references, maybe 15 to 40 studies analysed in detail, and then about 3 years to do a longer SR. So we're looking at sort of 25 to 40,000 references there, 100 to 150 studies analysed in detail. That takes quite a long time. And the problem we have is there are about 12,500 studies of bisphenol A now kind of indexed in the published literature. The number of studies has doubled since roughly 2014. And that's quite a lot of evidence to deal with. And there's a reflective of a broader trend in environmental health toxicological publishing, where in Medline, for example, we're seeing sort of 2,500 new studies indexed per day, right? So the amount of research that's been indexed in Medline has doubled since about 2012. So we've got this exponential growth in quantity of research being done, without necessarily exponential growth in the capacity that we have to deal with that research. And normally when we do systematic reviews, we have a keep it simple coping strategy that we use, where we kind of concentrate on single exposure outcome pairs to help us minimize data volume, resource requirements to do the research and the time that it takes. The problem with very, very focused reviews is that decision makers unfortunately have a lot of questions. And even if you can do your focused review in just 18 months, they really need answers to their questions right now, right? So they have wide-ranging information needs, they ask lots of questions, they're operating on urgent timelines, and many of the questions they have actually involve very large amounts of very complex data. So there's a poor fit between the best methods we have for making sense of evidence and the needs of the people who are actually using that evidence to make decisions. Which is kind of weird because we've got all this scientific evidence being created to support decision, and these days we're producing it at a greater and greater rate, probably never been more successful in terms of at least literature volume. But we're still only answering small numbers of narrowly focused questions whilst all this evidence is piling up. So evidence is effectively inaccessible to the decision-making processes that are funding it because of the time it takes to use it. And that's what we're really trying to address, and this is really what today's talks are all about. The reason that accessing scientific knowledge is so slow is that researchers, we're still publishing studies as PDF documents. So to get knowledge out of PDF documents, we, and I mean the royal we, if we get our postdocs to do this, have to read them and abstract that data from a document into a database. And that's fine if it's 1970 and there aren't that many documents, but it's not 2022, and there are literally millions and millions and millions of them, 28 million records in Medline. So we need new processes for making research data accessible and reusable at speed. So that's where our speakers today come in. So we've got Brian Alper from Computable Publishing LLC, who'll be talking to us about how ontologies help make research computable. We have Julie McMurray from University of Colorado Anschutz on how ontologies are built and how you can get involved. And we've got Dr. Wynand Alkema from Tenwise and University of Groningen on how to use ontologies in research. Each presentation will be 15 minutes long, and there'll be an audio signal for the speakers that sounds like this at 2 minutes. We'll have a 20-minute panel session to discuss your questions, so please do post questions in the Q&A or the chat box. This webinar is being recorded and it will be posted on our YouTube channel, which we'll share a link to. And the last thing I would say is please enjoy the presentations. I'd like to thank the speakers in advance. And Brian, I'm going to hand it over to you.
Okay, thank you. Share my slides here, and I'm happy to start off trying to answer the questions. What are ontologies and data standards, and why do we care? So just to start with some disclosures, I own Computable Publishing LLC, which develops software to support many of the things I'll be discussing, and includes a platform, the Fast Evidence Interoperability Resources, or FEVER platform, to support sharing all sorts of scientific knowledge in electronic standards for data exchange. I also have leadership roles in numerous nonprofit collaboratives, all related to these sorts of endeavors. So I come from the world of healthcare. I'm a physician by background, and in the healthcare knowledge ecosystem, we really want to generate evidence from all the data. We want to generate guidance. What should we do based on that evidence and using systematic review methodology? And ultimately, we want that guidance to facilitate or drive action to realize these effects in healthcare. And we'd like that action to reach back and get more data to understand the process and grow with it. But these different systems do not fit well with each other. And when we try to see this happen, it's like spinning our wheels. So ideally, the desired state of the healthcare knowledge ecosystem is each of these steps should interact well with all the others and be a continuous cycle with limited work to go from one phase to the next. We have experienced this in other parts of our lives. So let's consider when we travel from one location to another. At one time in the print world, we had these things called maps that would come out on pieces of paper. You could see the possible routes for your travel, but you were limited to the choices on the page. You could not in real time track where you were on the paper map, especially if you're driving at the same time. If the map was large enough, you might have to get out of the car to be able to unfold it and find the part of the map you want to explore. You couldn't possibly know what was happening, like a road closure or other things in the area in relation to where you were on that paper map. We have moved to the digital environment, and there we saw these PDFs, and you can get a lot more paper maps, thousands and thousands of paper map pages in one disk. And it was easier to get maps out for distribution, but you still couldn't use it when driving. You didn't have a computer to interact with that disk. And you'd have to stop and find the one you wanted to look at and then figure it out. But it would be easier than having to stop at every border crossing and get the local paper map. So the digital age helped with this navigation. We then came to experience the executable environment, the method and the tools where you can interact with the map. You can zoom in, you could highlight where you are, you could connect with other executable devices and software this GPS tracker could identify where you were in space and then coordinate with the map program. And as you traveled, you could see where you were on the map. Now, the GPS tracker device and software would have to match the map viewer device and software, or they wouldn't work with each other. And even if you had the specific GPS tracker and map navigator software working together, you still wouldn't know anything about road closures or where the local restaurants were or when they're open, because each approach would have to work with the software set up for that approach. We want to move to the computable era. Today, the knowledge is computable. While driving, a voice can tell you where to turn next if you miss your turn, and you'd ever have to actually see the map. You could know about available options in the neighborhood. Maybe you're interested in restaurants and what's open right now. Maybe a road closure happened. And the coordination between the GPS location software and the map viewing software and the local listings all seems to happen without extra efforts to make sure they're well integrated with each other. We could bring this power to the world of evidence. However, if we look at evidence, scientific knowledge, and the guidance derived from scientific knowledge, we still are very much thinking like a print world. Print is familiar and is conceptually how we organize much of our workflow. In the print world, the shareable value unit, what do we actually share with each other and transfer when we transfer knowledge, is a physical object. It's a relatively large unit with many bits of knowledge in one container. You know, we can physically share this folder, and there's many folders here, but each folder is a container with many bits of knowledge inside that folder. We then move on to the digital world. And this is the platform we use most today for dissemination of knowledge. We disseminate millions of PDFs as digital objects instead of millions of physical objects. But that PDF, right, might be a single article, the adverse effects of bisphenol A on male albino rats. It's still a relatively large unit. There's a lot of knowledge bits inside this article. We, of course, can use computers and get to an executable stage. And there are many specific software tools, but each tool is limited to the local execution, what is happening within that tool. Within the tool, you can share small digital objects, micro content. You could, if we have an example here, this is on PubMed, and we're looking at pieces of data from Medline, and we could isolate the title or the authors or the identifiers or the abstract and start to work with it in different ways and not require the entire object or record to be passed along. But if you want to try to work between this and a different tool, the software tools may not talk with each other, and you may have to re-enter the data. So we can do a lot more with executable tools, but now there's thousands of executable tools, and it gets complicated which ones to use and what happens when you want to use more than one. So ultimately, we want to reach the computable stage. This is when we have widely interactive, interoperable, integrated efforts across all of this. And this is the platform of the near future. We now can share the small digital objects or the entire PDF and large digital object with many small digital objects within it. But by sharing the small digital objects, it can enable us to select the data that we want based on the current context. The context could be our location, our reason for use, what tool we're using. We can customize how we view and work with that data. And we can make it easier to reuse the data and disseminate it instead of having to re-enter the data or do translation work to move the data from one system into another system. So what is needed to make that happen, right? We didn't get to the computable world for location data overnight. So there's 7 steps listed here. We need to agree to a common technical standard for this scientific data exchange. If we don't share it the same way, we can't understand it transferring from one system to another. With that technical standard, we then have the actual terminology that we share within the data, and a common standard vocabulary would make that easier. We then need to apply the standard and the vocabulary to our actual systems and our actual content. It can't just work in theory. We need to identify the challenges that get in the way of applying this to our systems and content, and then contribute change requests. Tell those who are developing the standard and the vocabulary what needs to be done to remove the barriers and make it better. Then we can use it and enjoy efficient support for scientific communication. We do all of this, we can shift our effort from processing tasks, re-entering the data, fussing with how to format the data just to work with the system you're working with, and put more of our efforts on adding value, creating new evidence, applying judgment to the evidence. So for the rest of this, I'm going to focus on the common standard vocabulary for scientific data exchange. What is a common standard vocabulary? Well, it's a standard if we have global agreement. And we need a human readable component. How do we understand this vocabulary? But as a technical standard, we need a machine readable component. How does a computer understand the vocabulary? So it could potentially be shared with different human readable displays for different audiences. So here is an example in computable code describing the attribute estimate for a particular statistic. We can see all the numbers, the level 95 or 95%, and the range from a low value to a high value. And within this, to understand that this is a 95% confidence interval, there's a coding element that has a display, a human readable component, a code, which is a machine readable component, and a system that defines that this code matches this display and perhaps includes definitions. What is this standard vocabulary? In this case, to describe the concept we call confidence interval. So one of the things we are developing now is the scientific evidence code system. And this is a screenshot with the term prevalence as we're developing this code system. We can zoom in here on the code, the preferred term, and the definition. The code is a machine readable element. In this case, we are working with a statistics ontology that has a code, stato, and this number. So we're reusing the code to help with interoperability across systems. The preferred term, prevalence, is a human readable representation of this concept. And then we have a definition, which is really rules or guidance when to apply this vocabulary term. So we can build vocabularies. We are doing it as I showed you for statistics in the scientific evidence code system, but there are also many vocabularies out there that we can adapt or continue to build and develop to describe populations, to describe exposures, to describe outcomes. You put all of this together and you can ask and answer complex questions like, what is the range of odds ratios reported across studies that are evaluating the effect of bisphenol A on fecundity in female rats? Well, female rats can be described as a population. Fecundity can be described in what does that exactly mean in precise terms as an outcome. We have the exposure, we have the statistics. We could even have study design terms. What type of studies are we selecting to answer this type of question? So standard vocabularies are needed for many concepts across such a complex question. So the scientific evidence code system is an open code system. No licenses will be required to be able to use it. And it can be applied to statistics, study design characteristics, and risk of bias of the study, whether based on design or based on application of the design. We are defining terms for standard expression of the risk of bias, both internal validity and external validity concerns related to the study. We can coordinate with similar developments for standard vocabularies developed for populations, for exposures, for outcomes. You put this all together and you can ask and answer many complex questions. So this is just showing the progress to date on the scientific evidence code system. And we have about 30% of nearly 600 or 535 terms now mapped out, and 30% of them have been fully approved by a global expert working group. Any disagreements or concerns, and we continue to deliberate. And that's the effort it takes to make a standard vocabulary that can be accepted by all. So we have a group, a virtual group called the COVID Knowledge Accelerator. This is really the manifestation of multiple global collaborative groups seeking to advance knowledge specific to COVID. And we were working with FHIR, Fast Healthcare Interoperability Resources, which is a standard for data exchange of health data. And we're extending FHIR to evidence and other resources to apply those standards for data exchange for scientific knowledge. So many groups seeking to advance COVID knowledge using this effort came together, and we created a virtual organization. No cost, no contracts. All meetings are open and transparent. You're welcome to join any of them. And it's a way to volunteer to accelerate the development and implementation of standards for evidence exchange. So we have a regular weekly meeting schedule. This is in Eastern time zone, tinyurl.com/COKA2021 if you want to see this. And you can see some of these working groups: terminology and ontology, research design working group, statistics, standard and terminology, risk of bias, terminology and tooling, specifically work on the scientific evidence code system that I showed you. So one way to understand this is, without a common standard, if there are 7 different systems wanting to talk with each other, you need to convert the data from one to the other in 42 different pathways. If we have a standard structured form for sharing all of this data, we only need 14 pathways to be able to communicate. You do not have to have a central hub, right? Any of these groups can talk with each other, but they will not have to develop specific ways to convert data for 42 combinations. And of course, we could provide a central hub to facilitate it as well. So that's what I have for you, and give back the rest of the time for others.
Brilliant. Thank you, Brian. That was a fantastic presentation. We're going to move quickly on to Julie now. So Julie, if you'd like to share your screen.
Okay, here I go.
Okay, we can see your screen and we can hear you. So
it says I'm unable to start my video. If someone could remedy that, please. Thanks.
Yeah, Camilla will take care of that.
Okay, so I will. Can everybody see?
Yep, we can see it.
So I'm going to be speaking on behalf of the Monarch Initiative. I am going to be focusing on the application of the technology, the ontologies specifically, rather than the creation of ontologies per se, as for mere mortals, 15 minutes wouldn't really be adequate to describe that. The link to these slides is here in the bottom. I will share that in chat a little later. So as has been described, ontologies are really super useful for different kinds of applications. Organizing data, this can be both prospectively organizing data and retrospectively curating published literature, for example. Filtering data, connecting data, and suggesting data. So what sort of vocabulary you need depends on the amount of semantic precision you need. And it should be said that the time and effort to build and maintain this kind of spectrum of vocabularies varies a lot more than the time and effort to simply choose terms and implement them. So really think carefully about what sort of questions you need to be able to answer at the end of the day. So when you think about an ontology, many of you have seen similar trees like this before, so I'm not going to dwell on it too much, but the idea is that the most general terms have subterms, which are more specific, and you get as granular as makes sense for that particular domain. There is no one-size-fits-all ontology for any application, and you can query terms across multiple ontologies in a variety of contexts, whether that's the EBI-OLS, which is my personal favorite, or BioPortal or Ontobee, and I'll give you the links for those sorts of things. But if you're not sure what sort of ontology you need for an application, poke around there and see what sort of terms are best represented. Once you have figured out the ontologies that make the most sense for you, think about how you're going to be deciding on the patterns about how you're going to be collecting that data specifically. And as I mentioned, you can mix and match from different ontologies depending on the level of precision that you need. I would recommend avoiding creating an ontology if you can help it. There are certain times when you just can't avoid it and you need to, be able to create a new ontology. But even in that situation, being able to cross-reference to ontology terms that may exist in a variety of other places is really important. So if we get down into the details of exposures per se, you can model an exposure according to the stimulus, the exposure event, and the receptor, together producing an outcome. And each of these has more granular components, and you can delve into the references there. So here's a little bit more detail here. So for a particular stimulus, there are a variety of sources that you can use to find terms that apply. You have the roots of absorption, the roots of exposure, whether that's absorption or injection, et cetera. And then the medium by which that exposure is encountered. So I also want to mention that if any part of the data collection process or the data annotation process that can be standardized is a love note to the future. It is making the most of that data in perpetuity. And so I just want to dive into a specific example here, and I will do a new share. Can folks see this spreadsheet?
Yes, we can see it.
Yeah, yes.
So this is an example of exposure to bendiocarb in zebrafish. And you'll notice the same receptor, stimulus, exposure event, outcome paradigm. And within the specifics here, I've used where possible available ontology terms. So for the species, we've used NCBI taxon. So while having the species is, we would say, required for this kind of metadata, NCBI taxon would be the source ontology we would recommend for that. And then likewise, having strain documented in the source and not just as free text somewhere in the body of a paper can be really helpful. And again, the exact ontology that you're going to use for a particular experiment will depend on all kinds of other factors. There's no one size fits all. But I thought it would be useful to just distill the practical implications of some of these more high-level conceptual things. And so, for example, for the life stage, there's the stage ontology for zebrafish. For the age, the stage ontology it could be appropriate, or for humans, there's also this ISO 8601 standard. And all of these are linked from the spreadsheet, and the spreadsheet is linked from the slides. So you can feel free to peruse it later. And we also welcome feedback. This is just a working example to just sort of highlight some of the possibilities and challenges associated with this kind of work. Here, we are just sort of speculating that you could have an experimental arm identifier, as well as an individual organism identifier. However, depending on the complexity of the experiment, it might make more sense to just say, we have this many animals, 30% of those animals in this arm develop such and such an outcome. So here for the stimulus identifier, we've got KEBI, which you all are well familiar with, I'm assuming. And then here's the ENVO for exposure in water, absorption via the EXO. And then those are put together as exposure to bendiocarb as an ectoterm. The dose concentration, dose time point or time points, attribution, and then we've just modeled the 2 time points that are in this particular paper. I've taken some liberties to show you kind of how this would be set up. The paper is not this explicit about the time points of observation. And so this is a challenge when you're going back to look at something that there's lossiness. And so creating clarity from the beginning is really important. Here's a different example, looking at aldiocarb, again in zebrafish, very similar. I just want to highlight sort of here, we have, for instance, a frequency of 30% in an experimental arm with 24 organisms. So I'm going to X out of here and go back to the presentation. Okay. Oops.
We can still see your screen fine, though we're looking at the spreadsheet at the moment.
Right. Let me X out of the spreadsheet so I can get to the slideshow and then do a new share.
So we're seeing a screenshot of your spreadsheet on a slide that has prospective data collection written at the top.
Okay.
Is that what we should be seeing?
That is what you should be seeing, yes.
Excellent.
Okay, great. As I've mentioned, it's also a love note to the past, making the most of the data that has already been published. But the fuzzy matching logic is considerably more complex when you're trying to integrate existing data. I should also mention that while data integration is a huge use case, 15 minutes doesn't even begin to provide enough time to go into that. And the tools available for integrating in a semantically aware way that data that is not inherently a graph is quite challenging. There are just gaps in there. This is going back a little bit to show why you would want to use ontology terms at all. So here's an example from the CZIT study at OSU, where we looked at how individuals characterize an observation. So one lab called it, for example, SOMI, another SOMITES distended, SOMITES undefined, SOMITE morphology in another lab. And so using ontologies in retrospective, we're able to harmonize them all to SOMITE morphology abnormal, which is a zebrafish phenotype term. That's obviously harder than doing it right from the beginning. So does that mean that everybody is going to use the same ontology term, even if they are annotating from the beginning? No, that's not the expectation. I mean, it'd be nice if they did, but different levels of granularity at a minimum are going to be implicated. And using the ontology consistently improves the, it improves the consistency across labs. So building and maintaining ontologies requires a lot of experience. I just want to caution folks who might be interested in building them, but anybody can find terms and use them. We have recently published a preprint in November on the PhenoPacket standard, and a new version of the standard has been released as of February. So if you haven't checked out PhenoPackets as a way to standardize this sort of case level phenotypic information sharing, please do. And I should mention this has primarily been developed for use in humans as case studies. However, it's our expectation that we're going to expand the functionality to include model organisms or support that a bit better. It is possible to use the PhenoPackets standard as it is now for model organism data, but it's just not quite as expressive or flexible as we'd like it to be. So stay tuned on that front. Here's what it would look like inside. We have a subject, a VCF file reference, specific phenotypes, and things like severity and onset, et cetera. So this is just a roundup of the open challenges and tooling gaps around the use of ontologies for this kind of work. So it would be great to have some real-time manuscript markup for phenotypes, diseases, genes, exposures, et cetera. So the best we've got is this sort of retrospective text mining. So there are a couple of options here linked that you could check out, where you put in the text and it tells you, hey, we think these are the following ontology terms that might be included. But there are all sorts of other tools. These are just a couple of examples. So spreadsheet autocomplete using ontology terms. There are a couple of things that that you can use for this. There isn't really a great tool for this right now for at least Google Sheets. There used to be Webulus, but this was now hamstrung by Google's throttling because it's a very IO intensive thing. So right field should still be working through Excel. And failing that, it is possible to just look up terms in OLS or BioPortal or Ontology. We don't yet have great tools for authoring PhenoPackets, but that's the goal. And one of the troubles that we constantly encounter is, okay, we've gone to the trouble of cleaning and structuring data, whether it's from a paper or something else. How do we share that transformation and cleaning so that others can make best use of it? So the best workaround currently is that the transformed data is made available through knowledge graphs and APIs and just word of mouth sharing, but there's not a great system yet for that. I also want to mention a new tool, which is called Schema Sheets, and it works to help you manage a schema data model for your experimental data. So I just learned about this yesterday, and so I don't have much to say about it, but there's a link here if you want to find out some more. And that's it. I just want to thank in particular Ann Thesson and Lauren Chan for helping to think through some of these tricky things, and all of the folks here that have been involved with the Monarch Initiative. Thanks.
That's brilliant. Thank you, Julie. That was a great presentation. We're going to move on again to Wynand. So Wynand, if you could try sharing your screen, make sure we've got that working. That is working, but I don't have any audio from you. Are you unmuted?
Am I still muted?
No, yeah, I can hear you now.
Okay. So I will, and I'm visible now as well, I guess, right?
You're visible now as well, yep.
Good, here we go. So I want to tell something about, so we've heard a lot about the value of ontologies and how you create ontologies. We are in fact at the rear end of the ontology process. We are using the ontologies in, let's say, a real world application. And then we'll show a little bit on how we use ontologies in helping people to search relevant literature when they do systematic reviews or scoping reviews. So I'm a CSO of Tenwise, a company that builds text mining solutions for the pharmaceutical industry and the food industry. Also professor of data science at the university, where we do a bit of research on these topics. So, already alluded to a little bit by Paul, systematic reviews. What do? You search a lot. You get an enormous set of documents. You filter those documents and you start to read a selected set. And based on what you read, what you find, you start to reason and you build knowledge in the end, which you publish in either a narrative review or a systematic review. And this is kind of the flow with an iterative part that you may optimize your keywords and your search phrases to cast the net a little bit wider or narrower, dependent on what you obtain with your searches. So how does text mining help in this process? That's shown over here. We can help in reading and in searching the papers that you are looking for. High throughput searching, we call that, and that's where we use a lot of these ontologies. But we can also help with text mining and the use of ontologies in filtering out your first document set, going from these thousands of documents to the final hundreds that you want to read. And in the end, we can also use ontologies to actually read the documents, to recognize concepts, to recognize relations between concepts. And that's more done with natural language processing, but of course has also a huge text mining component. And in all these phases, the keywords and the phrases that you use to search, filter, and that also triggers things, when you read those documents, they are represented by ontologies, at least the way we use it. And I want to show how that works in practice. I will draw from some examples from a tool that we have to demonstrate how these ontologies are being used in a real world application. So this really, I think, captures on the last bit of what Judy was also presenting. How do you find back knowledge? How do you find back concepts, genes, proteins, phenotypes, etc., in text that has already been published? And what we've done at Tenwise is to build a huge network based on a large number of ontologies that is available in the public domain. And we use all that information in those ontologies to scan through 35 million abstracts in PubMed. So we annotate all the abstracts with ontology terms, and also a large number of full text papers that we have access to. So in the end, using the ontology, using the, let's say, the keywords that describe those entities, we are able to build, and we call this knowledge maps. You see that on the right-hand side, knowledge maps that tell you how the terms in the ontology, represented by circles in this case in the map, how they are related to each other in the literature. And the idea is that if certain terms are often related to each other in literature, they are also related to each other in real life in the biology, because people publish on the relation between those ontology terms. I will draw from an example that I will refer to multiple times in this presentation, which is a bit related to skin diseases. We see here in orange, we see acne and skin aging and dermatitis as phenomenons of skin disease, basically. And in red, we see pathways that are related to those diseases. And in blue, we see the genes that are related to those diseases. And the nodes in that network are, in fact, referring to concepts that are drawn from an ontology. And I will show later on which ontologies we use, but because we use a structured vocabulary, the ontologies, we use a large set of Medline publications. We can click on any of these links in this knowledge network to see the underlying evidence that gave rise to a connection in this ontology-based network. For example, if we would click on the link between dermatitis on the right-hand side here in IL-22, we directly get all the papers, I'm only showing two here, where IL-22 and dermatitis are mentioned together in the abstract. And we also highlight these terms in the abstract so that it also facilitates screening and reading of that abstract. So, so I will mainly talk about genes and diseases and pathways, because that's where we use a lot the ontologies. And one of the most important ontologies that we use is maybe not really an ontology, more a structured vocabulary, but it's from the Human Genome Nomenclature Committee, and they have listed for almost 40,000 human genes. They have listed not only the approved symbol, like the example here, CCL17, but also the longer name, but also, and that's for us very important, the previous symbols or the other symbols that are being used, like TARC and ABCD2. So we use the ontology or the controlled vocabulary to harvest synonyms that people have been using previously in text to describe their gene or their protein. But this is a very authoritative resource that we can use. But also there, we still need to curate that a little bit. For example, one of the aliases or previous symbols that has been used for the gene TBXT is the symbol T, which if you do text mining with the symbol T will give you really a lot of hits. So also curated or semi-curated ontologies, you still have to, I would say, optimize a little bit for your specific use case. Normally, it wouldn't be a problem that you have a previous symbol like T if you're really interested in the function here. But if you do text mining, it is important. So, and that also goes back to Julie's talk that says always think about what is the ontology that I'm going to use, or the vocabulary, what is the use case, the research case, and that determines how you should use this, and what to take out of these ontologies and what to leave out. Another very important ontology that we use, and here we have the OLS system that was referred to earlier, is the human disease ontology. I'm just highlighting here the term dermatitis because we're talking about skin diseases. And what we see on the left-hand side is kind of the hierarchy of the term. So dermatitis is a child of, I hope this is visible for everybody. This may be a little bit small, but it's a child of the broader term skin disease. But on the other hand, we have also more narrow descriptions of the type of dermatitis that is that you can find in this classification. And what also is very important for us when we do text mining, we have the synonyms on the right-hand side here, eczema and skin inflammation, that according at least to this ontology is the same, an exact synonym as for dermatitis. So what we do in our applications, we harvest these keywords and these synonyms, and we search the entire Medline database to create those knowledge networks. And for us, it's very important to know that if we encounter the term eczema, it actually refers to dermatitis. And if we encounter skin inflammation, it also refers to dermatitis. So these three terms all refer to the same concept that we are interested in the literature. So the gene ontology, the human disease, the gene vocabulary, I should say, the human disease ontologies are very important for us. And thanks to, I think, a lot of people that put effort into curating these ontologies, we can build the applications on top of that. So this is just a screenshot of a web interface that we created based on PubMed and all our ontologies that we, or vocabularies that we have. You can search for the term dermatitis, and you are automatically presented with all the synonyms of this term as well. And you can select that term for investigating further. And what we see on the right-hand side are in fact, or is in fact the same data as I showed in the network before, only now structured table-wise. So we have the most related genes. In total, we have more than 2,000, but the most significant genes are listed here. That are related to dermatitis. The most related diseases, of course, dermatitis to itself, pathways, etc. So you can, based on the ontologies, you can map all the other concepts that are most related to dermatitis in the literature. And just making the link, let me check on the time, my 5 minutes, just making the link to the systematic reviews and literature finding. A lot of people use this system to get more ideas where they should be looking for in the literature. So they start with a certain term, but they get ideas for additional genes or diseases or other things that they should include in their search strategy. Also here we have, just as with the network, we have the underlying literature that connects basically the genes and the diseases. And what is important here, just highlighting that indeed, here we search with the concept dermatitis, but we retrieve the articles where, in fact, the synonym eczema was used, and also the term dermatitis here. So dermatitis, eczema is equivalent for us, and that allows us to aggregate a lot of search results into the same category, basically. The same goes for the genes. It's notoriously difficult to build a good vocabulary for genes because we have so many three-letter acronyms that could mean a gene name, but also could mean something completely different. Like CO2 is a gene name, but it's of course also carbon dioxide. So it's very important to curate that. But we see here the hits with CCL17, but we pick up also hits with the previous symbol TARC. So again, a good knowledge, having a synonym list that is very well curated and widely accepted, I should say, is crucial for harvesting these data. Last example that I want to give is not so much maybe an ontology issue, but more the knowledge on how people describe their pathways and their processes in biology. We know that a lot of people refer to signaling pathways as in this form, so the name of the protein or the proteins and signaling followed by pathway or not, but also people use just the term pathway or route or other things. So there's a lot of ways of referring to the same pathway. I would say it's not really a synonym, but often a different way of writing things down, with or without a hyphen, etc., with or without capitals. And that's also very important to incorporate that domain knowledge when developing your ontologies or your structured vocabularies. The other thing I want to highlight here is that because we text mine on a word level, we can also isolate where we have in which PubMed identifiers or which articles, we actually have a traceable statement of a pathway related to a disease. So we don't have to do this on an abstract level, but we can also do it on a sentence level. And this is also often used by people to quickly zoom into the even more relevant abstracts for their systematic or scoping or narrative reviews, because here actual, I would say, facts, actual relations between two concepts are being described. So yeah, well, I'm right on time, I think. I heard the sound. So I think the conclusion for this part of the talk, I would say without structured ontologies or controlled vocabularies or even glossaries, comprehensive searching is, I would say, not possible. And whenever you do a systematic review, you should, or a review in general for that matter, you should try to make use of those vocabularies. Sometimes you can use the public ones, and indeed already warned, do not try to make it yourself, but sometimes, and we feel for our applications often, we need to create our own, I wouldn't say ontology, but our own controlled vocabularies to change the way terms are defined into a way that people describe it in text. Because saying it in a formalized manner is another thing than using it in scientific text. People use different terms there. And this second bullet should be guided, again, reiterating some things that already have been said, by expert domain knowledge, but also the specific use case that you're after. Sometimes some parts of an ontology are important, or some properties of an ontology are important for one use case that are not really important for another one. So keep that in mind when trying to use ontologies or controlled vocabularies for that matter. Yeah, I'll leave it at this. And I think all the questions will be taken in the Q&A that's coming up.
That's fantastic. Thank you, Wynand. That was a great talk. Yeah, so if I could just invite the speakers, sorry, to just turn your cameras on so we can see you all. Yeah, so we had a few questions come in from the attendees. So Accelerator, I have to apologise if I almost certainly pronounce it wrong, is asking saying that he seems to him that one of the big difficulties in any such efforts of knowledge representation is getting agreement on the terminology definition and the hierarchical linkages, right? So this is a philosophical matter, the question of whether we can unambiguously describe some domain that we can all agree on can sometimes be a tough hurdle. I think probably Brian, you might want to speak to that first, and then I'd invite either Wynand or Julie to respond as well.
Sure. Well, first, thanks for the comment, and I agree. It is a challenge, And for the scientific evidence code system, this is open for anyone to participate, and we have global participation. So to reach agreement on the terms, we have comments saying that one word isn't quite right for the definition, and we go back and adjust further. Or it's not really where you put it in the hierarchy. And sometimes we end up making an adjustment to the hierarchy and have to go back and change, 10 or 20 other terms to fit within the hierarchy as we do it. So it's a complex, language is complex in the as well. So trying to get it to be exact and understood in so many ways is a complex phenomenon, and it's naturally going to take a fair amount of effort to get agreement from the multiple perspectives.
Yeah, Julie or Wynand, did either of you have any thoughts on that question as well?
We have tried this knowledge representation in various projects, and I think that the same goes as for ontology development. You can debate for years on what the right hierarchy is and what the right level should be. We often, well, at a certain point, try it the other way around. We just start, and we only, with representing the bits of knowledge that we are interested in, and only if we feel that our terms are not specific enough for a certain aspect of that knowledge, we branch into more specific terms. So we just let the process guide where we need to explore more and where it is needed, rather than trying to develop that all up front. And that works in practice okay, I would say.
I second that heartily. I specifically recommend a practice which we call competency queries, which is that you think hard about the sorts of questions you need to be able to answer at the end. And that's step 0. Yeah. And only from there can you go back and start to model that knowledge in a way that makes sense, because these things are what we call endlessly perfectible. And you can really get yourself down the rabbit hole.
So I guess the lesson is that developing controlled vocabularies and taxonomies is challenging. So view it as a process for solving a task or a problem, not just something to kind of pursue as a hobby, right?
Right.
Yeah, so I think one of the things that kind of came up, and maybe was kind of a subtext of the chat, is that, when we're developing these taxonomies, right, we're kind of defining terms in a way that kind of as many people as possible agree on. But then we have terms and definitions that people then have to use. And that can be, sort of implies quite a disciplined and structured way of speaking, right? So whilst we might be able to agree these definitions and agree these terms, how then do we get people using these terms in a consistent and agreed upon manner? Which, because I could imagine some people saying, yeah, but this all sounds very good, but science isn't rigid and communication isn't rigid. All these systems you're building for vocabulary use, they're very constraining. So how do we help communicate the value of such tools and things to researchers, do you think? I think, Julie, if we start with you, since we have you, and then.
I think it's helpful to bound expectations about what can be agreed upon, because one of the strengths of ontologies is that they're able to bridge some heterogeneity. And while it's important to agree on the definition of terms, it's less important to agree on the full implementation, right? One group might want to annotate data at a much more granular level, and that's okay. That's what ontologies are for. And so the perfect is definitely the enemy of the good. That's what I would say. And to find, basically define the smallest possible unit that needs to agree in order for things to work.
Yeah. Brian, because we're obviously working together on developing Sevco, so do you have any thoughts on that?
Yeah, well, I'll say in theory and practice are the same. In practice, they are not. So what I mean by that is when you're trying to develop an ontology, you can take a theoretical approach and everyone could be focused on being exactly correct. and meaningful in their worldview of how this comes together, or we can focus on a practical application and what will be useful in this context. If you try to do both, you can have the endless debates because neither is necessarily right or wrong. There are different ways of approaching it, and you cannot always achieve both of those models. So context matters. And if you are doing it for the context of application, frequent reminders to the group of the context, and then putting things through that context filter as you determine and develop it will lead to different results. And if the context is annotation by machine learning on a vast scale, you need a different level of granularity than if the context is application by humans from selecting a small number of terms. So you have to be very context aware to make it manageable and keep people on track.
Wynand, I saw you nodding very enthusiastically a couple of times there. So if you want to...
Yeah, I have nothing to add here to it. Fully agree.
Brilliant. So I think so. Oh, yeah.
Sorry, could I just add one more quick thing? And that is to say that the conceptual alignment is important, but it's possible to have preferred labels for different contexts. And that's something we've embraced, for example, in the Mondo disease ontology, that there are certain cases where, for instance, in a lay population, you might want something that's less clinically oriented, but it's the same thing as a clinical term. Those sorts of things are context-aware specifications are something ontologies are really good at, and we should leverage as much as possible.
Right. And you had a great slide where you showed the progression from glossary all the way to ontology. And ontology is the most complex way of putting it all together. So we're actually somewhere in the middle, the standard vocabulary with the code system we're working on. But it comes up as people want things that are like an ontology, and we're trying to be supportive of future ontology development. We're addressing what we can, but trying not to have to build all the different context-specific preferred terms in the path and things like that. So one of the things to pay attention to is how complex and do you really need to go all the way to an ontology to meet your focused goal, and how much of this do you need to build to get the level of standard vocabulary that you're looking for?
Mm-hmm.
Katya, you have your hand up.
Yeah, I just want to speak up for bench scientists, where I spent beginning 15 years of my career. And how do we marry or accommodate for scientific creativity and this, right? Because it could be perceived as the straitjacket that you all want to put on us on top of everything else, the other hoops that we have to jump through. Also, we have to say it in this specific way, right? In that, and describe like this. That's why I'm sort of advocating for this global open dictionary that can translate. Of course, the harmonization is very useful. I'm totally on board with this. But there are some pieces of, especially the primary research, that will inevitably either be unused if they're undecipherable, or be basically forgotten, or So we need to have some sort of a system where both worlds can kind of exist.
So yes, one set of tools that we've developed is called the SSOM model, SSSOM, which is the simple standard for sharing ontology mappings. So you might have an ontology, and I might have an ontology, and I might want to cross-reference your terms, but you may do that sort of reverse method in a slightly different way. And so being able to say, here's what I think is an equivalent term, or a more narrow term, or an exact match, and being able to do that in a lightweight way is really quite helpful. It's not a global dictionary. There will never be such a thing. but understanding the provenance of what terms are deemed equivalent could be very powerful. So I'll put the link for that in chat.
Yeah, and providing some sort of a reward system for the folks to use it so that their papers are more visible, it's more used, or what have you.
So there are many tools that I know EBI has one. I think it's What Is It, but where you can put in fact your text, and it tries to annotate this with ontology terms. And I could well envision that for example, publishers or at the moment of submission, you submit your abstract, and people are going to make, based on these tools, some suggestions for rewording your research. So it's still up to you whether you want to rephrase, but suggest the correct G names, suggest the correct disease names, etc., etc. That could already be very, very helpful, at least for mining the research.
So I think so. Joanne Danebostel has just dropped into the chat saying that basic research is more likely to be used and appreciated if it can be found through use of a standard vocabulary, right? I think, Julie, one of your slides, so your spreadsheet was actually, I think, quite illuminating, because it actually, to me at least, as like an editor of research, right, showed that ontologies can actually act as kind of sort of de facto reporting standards, right? You have a set of fields that we know are important for people describing their research, and then a set of standard terms that you are helped to populate those fields with, that are then, because they're machine readable, will be more readily accessible by research databases, and it will make your research more discoverable. So I don't know if you have any thoughts on that specifically.
Absolutely. And you know, one of the main takeaways I'd like this group to get is that we shouldn't wither in fear of not being able to make these perfect. There is no such thing as perfect. So the more ontology terms you can incorporate into your general reporting workflow, the better. Even if that ontology term you've chosen belongs to an ontology that goes into disrepair in 20 years, it's still better than some free text term. I still recommend both the ID and the label be used in combination, just in case the ontology fails to be maintained or the preferred label changes or what have you. Understanding what was in the mind of the person annotating is an important component. But being able to transform that data in a readily, you know, basically make a knowledge graph out of some experimental data would be possible if those nodes are labeled, right? If you just have AB as a strain in a field, that could be antibody, it could be a zebrafish strain, it could be any number of things. And so by being explicit, you make the job of the computability so much easier.
Ryan, I think you probably will have something to say on this as well.
Sure. Just to give an analogy, I think many here could relate to if you've presented at conferences and you're asked when filling out the forms online, associate some keywords with your presentation. And one form may just give you type in anything and it will be added as the keywords. A different one may give you a fixed list and you must choose from these and pick whatever you think applies. And another one may give you the list, but you can also add terms. Now, why are you doing this? So people can find your presentation in whatever search, you know, comes later when they're looking for things. But which of those would you prefer? If you have the list you can choose from, you don't have to make up terms that people may or may not use, but you have a specialty in whatever you're presenting, so you want to be able to add the terms there as well. So Katja, you were talking about the need for an open approach. Don't force people to be restricted to the ontologies, right? Use the ontologies when they're there, so you don't have to reinvent terms and it makes it easier for people to find, but be able to add. Maybe the terms can later get added to the ontologies as more people use it and need a definition for it. But ideally, we should have systems that support both. And there's ways in the technical design to say the choices are extensible, meaning use from available choices when you can and be able to add the check the box for other and write in answers when you do not find something that is already there.
Right, thank you, Brian. So we're going to take another question from the audience. We have someone asking, what role do subject matter experts play in curation, and what is best practice for engaging them? So I think, Wynand, if you want to maybe start with the answer to that, and if you could also just help maybe those in the audience who have maybe don't know what curation is, start by defining that, and then that'll be great.
Oh, the question is what the role is of the subject matter experts. I think it's almost a crucial one, because they often know best what is really meant by a term. For example, I know a bit of immunology and signaling, so I know that if people say signaling and pathway, they actually mean the same. Or when they use a hyphen in between two gene names, it means that there's an interaction between two genes. These kind of things you just know and are important for developing that. So also subject matter experts can say it's easy to merge these two terms, because they practically in daily work mean the same. But I don't know. On kinase research, it's very important to discriminate between the one binding pocket and the other binding pocket. So you should have special terms for that, because we know that from our drug development expertise. So yeah, I think it's the most important a role, in fact, to guide, to evaluate whether terms should be included, merged, or disjoined in these ontologies. Yeah. So, for example, we are making a controlled vocabulary for metabolites in biological research. If we download a database with the word, I don't know, electron, we know that it's not the most obvious term to include, because that's not how people often talk about metabolites in enzyme metapathways, etc. So yeah, there's lots to do for subject matter experts in that respect.
Stuff. Brian, if you want to respond.
Yeah, so definitely subject matter experts are useful for curation or making sure the stuff is right, because you may not understand nuances in the field, that the definition has a different meaning among those in the field and how they use the term. And when you go to apply it to real data, what will it mean and what will it capture? At the same time, if you are focused on an application ontology, it's important to have the people who are expert in what it means to them representing the people who will use the vocabulary. So if the vocabulary is completely created by the experts, and it makes sense if you have all the expert training, but then people don't know what it means in another context, you may not meet the goals of why you were setting up the vocabulary. I recognize, in medicine, an outcome could be vertebral fracture. And if you have this exposure and it increases the rate of vertebral fracture in postmenopausal women, for example, that can sound terrible, right? It literally can mean broken back, which sounds like it'd be a very painful, debilitating situation. But technically, based on how it's used in the literature, X-ray screening that finds changes in the structure of the bone, the vertebra, the spine, would also be called the vertebral fracture, and it may not cause any symptoms or problems. So you need both expertise in what does the term mean, but also what does it mean in context by someone who's going to use the term, or you could get, odd results that are technically correct, but misleading.
Very good, thank you. And Julie, we're going to give you the final word before we then wrap up.
Oh, context is all. And knowing the audience is one of the most important things in considering how to develop an ontology, how to apply the ontology, and how to query terms, or how to query data that's been annotated using the ontology. But even given all of these challenges, ontologies are still usually the best way to overcome the heterogeneity and granularity differences in the data. So thanks, everybody.
That's great. Thank you very much. So I'd like to just say thank you to our three speakers, so Wynand, Brian, Julie. They were great talks. I hope everybody who took part, attended, enjoyed them and found them informative. I will be uploading this presentation to YouTube in the next week or so. Katya, I don't know if you want to give people information about how to get in touch with us.
Yes, thank you so much. I did put a link at the beginning to how to subscribe to the newsletter of EBTC. We can put it in again. And also thank you very much to the speakers and to the attendees for invigorating discussion. And we'll keep working together. It's a tough problem. Thank you.
Thank you, everyone, and goodbye.